Asymmetrics, talking about his stem cell counting technology.
Good afternoon. I want to first start by thanking Kyle and the organizers for an opportunity to come and talk with you today. This is my third meeting, and I really have enjoyed coming here. And it's been a real support for us to have an opportunity to talk about the developments that we're trying to bring into the industry with this particular group. And so with that, what I want to do today is to tell you about our most recent advance. And this is, in fact, the first time I've given a public presentation on it, so I'm really quite looking forward to this. So here's the outline for the talk today. I want to start off by reviewing tissue stem cells, getting us all on the same page, and reintroduce again their unique counting problem. I then want to tell you about Asymmetric's first-in-kind solution, which is a computational simulation solution to solving that problem. And I want to get a little bit more in depth this time so that you have a better sense of exactly what we're doing as opposed to focusing on the output. And then I'll close with the recent advance, which is a new way to count stem cells quickly based on simple doubling time data. So to begin with, this is the cell that I want to talk about today. And this diagram here captures the idea that this is a cell that has duality in its function. And that duality exists in many different places. So these tissue stem cells can be found in the hematopoietic system. They're epithelial, they're endothelial, they're mesenchymal cells. And for this audience, it's a bunch of cells that are tissue stem cells in the perinatal tissues. And the thing I'd like for you to take away today is that even though they're all in these different places, they have this one unique thing in common, and that is the thing which names them. It is the mnemonic for tissue stem cells, and that is asymmetric self-renewal. It is this remarkable ability of these cells to undergo division events, make two cells, where one of the cells is essentially like the starting cell. And the other one is a differentiated lineage cell that goes off and makes whatever tissue this stem cell happens to reside in. Now, I'll just share that this particular diagram I have here, over the years in the literature in this field, there's been some debate over how the cell accomplishes asymmetric self-renewal. There's this deterministic idea where single cells can divide and make a cell that's different and a cell that's the same. And there's also these ideas that, in fact, this happens in compartments where stem cells are doing two different things. Sometimes they're dividing and making more stem cells, and sometimes they're dividing and making something different. But for our purposes and for our method, it doesn't matter how they do it. What's important is that they do this, that they have this ability to divide and not change in their phenotypic properties while giving rise to cells that make all the functional parts of our bodies. Now, as I said, doesn't matter for the counting problem. The counting problem derives from the essence of the cell. It's asymmetric self-renewal, and I want to take you through that. So this schematic here shows what's going on back in tissues. Adult tissue stem cells, be they perinatal or postnatal, undergo these asymmetric self-renewal divisions to maintain their state and their fraction. And that fraction is low compared to all the cells which derive from it. So there are these transiently amplifying, mini-amplifying divisions that are called committed progenitor cells, which then go on to make cells which undergo the full differentiation. So if you're in liver, these are hepatocytes. If you're in the intestines, these are the transport cells in the intestines. So Mother Nature made vertebrates have this breakdown between all of these cells that we have to produce to exist and the cells that we need to maintain that existence. So the stem cells are responsible for maintenance, and they're also responsible for genetic fidelity, maintaining the genome. While these cells right here, they are produced and they turn over. They're being lost all the time. And what's important to note is that there's no backwards arrow here. These are the cells that do this, these cells do not. And that's important for treatments where you need long-term curative therapies. If you give committed progenitors, you can get some effects, but you will not get long-term effects on the span of the lifespan of the patient. That requires the stem cell. Now, the problem with counting begins with the biology of the stem cell because it's rare in tissues. As I showed you, there are lots of other cells that are being produced by this cell, but its number is small. And that difference here in tissues continues in isolation preparations. Stem cell populations are not homogeneous. Ruspa gave a very good introduction to what I'm telling you now. I'm re-emphasizing it. They are heterogeneous. And importantly, the critical factor for long-term care is a small fraction of the population. That's the first problem that makes them difficult to count. The second problem Ruspa also referred to is this problem that we have right now, it may change, that there are no stem cell specific biomarkers, no specific molecular biomarkers. The ones we've been talking about here that you know, like CD34, CD133, and CD90, they are expressed on stem cells. The problem for counting is that they're also expressed on many of the committed progenitors that are the progeny of stem cells that outnumber stem cells in vivo, and they certainly outnumber cells when they're isolated. They also are a problem when we do expansion, because, though I won't have time to go into this today, because of the asymmetric self-renewal, unless a stem cell divides symmetrically, which it can at some frequency, its number goes down with time as you make more and more cells. We'll come back to that point when I talk with you about our counting technology. So, when I started this company, I was of the view, having been in the field for most of my career, that everybody pretty much understood these ideas. The world's a bigger place than just my laboratory, as I've found. And so we've been trying in our company to get a better sense in the world of what's the landscape of knowledge about stem cells. You may have seen this if you've gone to our website, because we basically ask people to come to the site and answer a few questions about their stem cell knowledge. And I want to share some of that with you because it's been very enlightening for me. So this is an ongoing public survey. Our numbers are kind of small now. N is 75. I'd like for that to be about maybe 4,000 or 5,000. We're working on it. Send your friends to the site. But I want to point out that about half of those respondents have identified themselves as being academic and industry professionals in the area of stem cells. And these are the kinds of responses we're getting. So of that group, 37% believe that stem cell preparations are homogeneous, and they are not. About half of the people have responded that they understand that there are no stem cell specific biomarkers. That's good to see for companies like mine. The problem is that 80% think the stem cells can be counted now. And they think it's done by flow cytometry, and you cannot count stem cells by flow cytometry because there are no specific biomarkers. That's the problem we're facing now with our counting technology. 22% believe the FDA requires that you know your stem cell dose specifically. And the FDA doesn't require that. Not yet, anyway. But I want to leave with this question that we've asked that we got an answer to. 80% believe that stem cell specific dose, knowing the number of stem cells, is important for clinical trials, to design them well and to interpret them well. And so we build on that idea because that's why we think we should be doing what we're doing. So that's a lead-in to say that there are a lot of unmet needs for stem cell counting. In academic research, knowing how many stem cells are in your experiments. As I just mentioned in therapeutics, knowing the specific, if you think the stem cell is important for the treatment outcome, how can we be doing medicine without knowing the specific dose? How can we interpret trials without knowing the specific dose? Gene therapies where stem cells are being modified, how many stem cells are modified? How many modified stem cells did the patient get? In biomanufacturing, how do you optimize production if you think the stem cells are important if you're not quantifying it? And even in drug development, there are places where knowing something about stem cells, because of their role in the function of our bodies, knowing the specific dose and the specific quantity would be important. So because of this idea, I got really excited about this. This is last year. This is the FDA, its standards coordinating body, has this new document that they were drafting, this is a draft, of needed standards, standards that are needed to advance regenerative medicine. And for the first time, this problem of unknown stem cell specific dose was addressed. So when I got ready for this meeting, I thought I should probably update this. I don't want to show up with a draft. I want to have the thing that was in the report that came out in January of this year, and I couldn't find it. It's been replaced by C21. C21 is unknown cell-specific doses. Stem cell-specific dose and stem cell dose does not appear in this entire document. I don't know why it's gone, but it is a problem that it's gone. And you see, these are the challenges we have with this idea in the industry. What I want to submit to you today is that this problem is not going to go away by us ignoring it. And so for the past 6 years in Asymmetrics, we've been trying to figure out a way to address the problem and to educate the industry about the importance of it. So with that, I want to turn to telling you about the solution and dig a little bit deeper into how we do this. So with our partner company AlphaStar, which is a software engineering company, we put together these ideas of stem cell kinetic modeling, and we have this new test we call the AlphaSim Test that allows us to count tissue stem cells from any tissue specifically for the first time. I want to tell you about how we do that. So we start with these ideas. This is our translated biology. We're going to do computer simulation based on the understanding that this is how cells not only grow in the body, but how they continue to grow in the test tube. That stem cells divide asymmetrically primarily. They can sometimes divide symmetrically. We'll talk about that. But they give rise to cells which ultimately produce cells that stop dividing in culture. This is their default state. And the way we've gone about getting access to that biology is by something called serial culturing. So what we do, if you were to bring a sample, if you had a sample you wanted to count, it doesn't matter whether they're adherent cells or suspended cells, we put them in culture. We let them grow for a finite amount of time, and then we count them. We take a fraction, we passage a fraction to a new dish, and we do that process over and over again, iteratively. And by doing that and keeping track of the cell counts, we can keep track of how many times that culture doubles with time. And we get these plots that are called cumulative population doubling curves. This is a schematic data with time and the number of population doublings. In all human tissues, there are no exceptions, and you hardly ever get to say that in biology, they all will stop dividing. There's debate over why they stop dividing, but that's a feature of why we're able to count stem cells, which are the only cells in the system that can divide indefinitely, and these divisions are dependent on their number. So we take this data, in this case here, this is actually 6 independent cumulative population doubling curves. We model it with these ideas, and we use that to find out how many stem cells are responsible for this. I want to tell you about how we do that. So we have the idea that cumulative population doubling, that data, could be defined by some function, some algorithm that involve both things that we can measure that were important and the things we want to know, which we don't know, these unknown factors such as stem cell number. There are a lot of factors here, right? So we have all these measured factors that we can measure and put into our model. And then we have all of these unknowns that we're trying to find. And we have to find them simultaneously. And that's where our computing has really been important. I want to give you a sense of what that computing is like, right? Because later on when I tell you about the new advance, you have to trust me that the computing actually, not trust me, see that the computing really is powerful as I say it is. All right, so how do you match all of these? Well, here's what we're doing. We have data. And we have a model that tells the computer, how do you calculate how many cells are going to be produced over time with all of these factors put in? We put in the factors that we know. We give the computer those. And then there are these factors here that the computer has to find. We let it randomly sample numbers and throw them in and simulate the data, compute the output. And then we compare it to the real data. And we ask, how close are we? And if we can find that we can get very close to the real data, then we have these parameters. So I want to show you how the computer does that. This is a machine learning program, and what I'm showing you here is the computer doing a sampling, computing, matching to the data, and asking how good is the match. This score gets smaller and smaller when the match gets better and better. The computer program we wrote is machine learning. It remembers. So it's keeping track of its low scores. That's what that black line is. While it keeps looking. And when it gets a better one, it keeps track of it. And then we get down here and we get some score that's very low, and we then can ask, how specific is this set of parameters that we found? And it turns out it's very special, because this is all of those scores that we got for that particular search, which was 1,000 iterations. And this is the score that we end up with. It's important that there are not many solutions. These solutions are exact within our experimental error. And that means that when I do this over and over again, which is what I do when we generate data, I get the same set every time around, like this. Okay? So in that set is the stem cell specific number. We'll talk more about that now. So again, to summarize, we take data, we do a computer simulation to find things that can define it. This is an example of that best fit set of factors simulating this data. It's a pretty good simulation. And if you take the mean of these simulated data and the mean of the real data, they compare very well. So we input measured factors, we let the computer find the unknowns that we want. And once we've done that, we can do something that's really neat in simulation space. You can deconstruct, and now you can evaluate any one of those factors, either the ones you measured or the ones you found, and ask, how do they change during this course? And of course, what we want to know is, how does the stem cell number change during this course? This is our very first stem cell count. I showed this slide 3 meetings ago. I want to blow it up a little bit. So we're looking at, during that serial passaging, the number of stem cells that are being produced in that culture over time. These vertical drops here are when we split the culture, right? So I want to just say that this feature that you see, we didn't build it into the model. It comes out of the model. That with serial culture of any human cells we've looked at so far, stem cell number decreases with serial passage. It's not surprising that phenotypes that are associated with stem cells are lost with passage with time, because the stem cells have been lost. I just want to give you some examples of this, because this is expanded liver. These are bone marrow hematopoietic cells. These are bone marrow mesenchymal cells. And something I want you to take away from this, though. We learned something in the simulation that I didn't expect. I didn't even think about it until I saw it. It's like, why are these things flat and these things pointy? Right? What we're looking at here is, this was the stem cell number at the start of the culture, and the stem cell number didn't change during that growth period, because those cells are growing 100% asymmetrically. They're producing other cells, but their number's not changing. And we see that for bone marrow and bone marrow mesenchymal cells that everybody knows are really, really hard to expand. In contrast, these liver-derived stem cells, you see these vertical inclines before the split. Those are symmetric divisions. Those are stem cells making more stem cells. They still end up being declined because their rate can't keep up with our splitting. But this here shows that it's possible to see stem cells are actually dividing symmetrically as well. We capture both of them, as I said earlier. It doesn't matter whether they're doing one or the other, we capture them both. One other thing I'll just say quickly is that we can solve that other problem kinetically, because we can tell the difference between the stem cells shown here in blue during one of these serial culture series, and these guys right here are the non-dividing cells produced in the culture over time. Notice at the end of the culture, the non-dividing cells are what we know biologically. Nothing's dividing at the end. And this green trace here is both the transiently amplifying cells and the non-dividing cells. So if you look at the area in between the two, you get to see what the transient amplifying cells are doing. And at the end of the experiment, there are none of those. They're all non-dividing cells, which is what we observe biologically. The program captures that. So using this over the past couple of years, we've been able to show that we can count a lot of different stem cells. We've done some validation in many of these compartments. Of course, for this audience here, umbilical cord blood stem cells and mesenchymal cells are of interest, and also amniotic membrane. I'm going to focus on these two types of cells going forward. Okay? So I want to give you some sense of the kind of output we do get, a little bit of validation thrown in as well. And what I'm going to be showing you is from this analysis that I've told you, where we have these things that we know and the things that we found, these were discovered. I'm going to show you these, what we call stem cell kinetic programs now. This is a prediction of what these cells are doing in the culture system, starting with time 0. So this was one of our first validation analyses. This is human CD34 bone marrow cells, and we're interested in the hematopoietic stem cell count. And what you see here is that our profile shows that that number is approximately 3 in 10,000 cells. You can hear that number thrown around today between 1 or so and 10,000. It's about 3. This is what you get in the literature on average for limiting dilution skid mouse or population cell assays, which is the best estimate prior to this work for how many hematopoietic stem cells there are in bone marrow. So we're very close to that number. And then going forward, this is a comparison here that starts to get into some validation. So those biomarkers like CD34, which don't identify stem cells specifically, can still be used to manipulate stem cells, to move them around, to enrich them. So this is not a congruent study, but this is looking at whole cord blood and CD34 enriched cells from cord blood. And the number that we see for whole cord blood is about 33 stem cells per 10,000. It's about a 20-fold increase when you do a preparation that's been enriched for CD34 cells, which also enriches for hematopoietic stem cells. I want to point out one other thing about this slide. This number right here tells us whether or not the cells are growing symmetrically. I showed you earlier for bone marrow, and the same thing's true for whole cord blood. These cells barely ever divide symmetrically. They dilute very rapidly in culture. But this is 27%. One in every 3 cells in this population is dividing symmetrically. And one of the reasons we think this is true is because the stem cells are concentrated. And some of the work in my academic days shows that when stem cells are at higher density, they show more symmetric division. And I think that's probably what's going on there. Now, I just want to highlight that a little bit more so you can see it in the data. This is the whole cord blood. There are actually 2 independent studies done here. You see this decline in stem cells with these flat points because there's no symmetric division. And here with the cord blood that's been enriched for CD34, you still get that rapid decline, but you see these increases here that represent that 1 in 3 cells dividing symmetrically every cycle. And then this study here is from the literature, but it's nice because it's the same starting preparation fractionated into CD34 plus and CD34 minus cells. And for the minus cells, we barely detect any stem cells at all. That's at our level of detection there. And you see here for the CD34 positive cells, it's about a 20-fold increase when you do that fractionation directly. And again, for these enriched cells, you see this high rate of symmetric division. I want to close with this example before getting to the new data of umbilical cord mesenchymal cells. This is 2 independent preparations from 2 different groups. And what you see is that for these mesenchymal cells, there is a much higher stem cell fraction. This is about 5% stem cells. And this is what we're finding is consistently true with these mesenchymal preparations, except for bone marrow. This is 12% stem cells here, right? The other thing about these cultures that's exciting, to me at least, is that this is 20% symmetric division. This is 35%, right? So the thing I want to drive home today is that if you took these 2 preparations and you use them in your clinical study, you might say we have the same number of stem cells, 10,000 here and 10,000 there. Those samples may be very different in terms of what they do for patients because there's about 2.5 times more cells here that are stem cells. If the stem cells are important, that's going to matter. Getting back to the symmetric division, if you're doing studies trying to figure out ways to expand stem cells, you need to make them grow symmetrically. You got to ask whether or not your agent is increasing that number. And that's something that we can now score with this method. Okay, we've been very excited, you can hear it in my voice about this, but there are challenges. The method has challenges. And I'm listing them here because that's what I want to talk about now and close with. Time. If you didn't take it away yet from those slides I've shown you, it takes us right now, our best, is about 3 to 4 weeks You give me a sample today, or you start a sample today, it'll be 3 to 4 weeks before you have stem cell count. And there are a whole lot of reasons why that's just really a problem for a really good tool. There are also counting errors. I put this in parentheses, not because we make errors, but because we also now have relationships with academic groups where they do all of the serial analysis. They send us the data, and we do the analysis for them. And right now, by the way, we're doing that in collaborations to no charge. The problem is that that's a lot of counting over time, and we're finding that errors get made. So it's an error prone. And then it's expensive because there's all of this cell culture going on, and those of you who've done it know, even when you're working with something like fetal calf serum, it's expensive, but go to purified growth factors, and it's astronomical. So we need to make it faster, and we need to reduce the cost. And I'm going to tell you about how I think we've done that. So this is the slide I started off with. I told you about the fact that there ought to be some function here, some algorithm. We couldn't even imagine how to put all these things together into that, although I had been thinking about it. I couldn't figure it out. So simulations solved that problem. But now we're at a new place. We started out with measured factors and unknowns. Now we have measured factors, and we have discovered these other factors. I still couldn't figure out how to write an algorithm. But it occurs to us that there should be some function of a simple 3-day or 2-day population doubling study. Just grow cells for 2 days, determine how much they doubled. That tells you the stem cell fraction. And what I want to tell you now is that we have found such algorithms. And the answer was in the data. I've been showing you these types of plots here where stem cell number with time. And I want to say something else too. I've been saying this to people because I think it's true. If I had been in an academic laboratory with talented people like Ruspa Takizadeh, we would have known this 3 years ago, because students would have seen it. I didn't. And when you see this, you're going to say, gee whiz, how'd you guys miss this? All right, so we can also look at not the number, but the percentage of stem cells over time. I showed you one of these traces earlier. We know the total cell number at every point, so we can calculate the percent. But there's a piece of data, we don't put it in our reports, I don't think about it anymore, that turns out to be pretty important. It's this data set here. This is the change in total cell number over time. This is the computer simulation. But I just want to tell you, the computer is simulating something that we actually measure. So think about what a serial culture is like. You put cells in culture, they grow. In this case here, in the early phases, they fill up the dish, they can't grow anymore, so they stop growing there. Okay, and then we come along, we count them, we know that number, and then we split them, and we know this number. During the next period, they grow again. We know the new number. If you know two differences in cell number and time, you know the population doubling time. So this is simulated, but we also actually know it in real life. So we know the population doubling time as a function of stem cell fraction. Interrogating that relationship, if there is a function there, we would discover it. So that's the basic idea. Relate this population doubling time, which we can calculate, to the stem cell fraction that we have found with our powerful method. We can find this function, and I'm going to show it to you now. They look like this. This is 3 independent studies with different tissues for stem cells. Some of them I've shown you. And we're looking at the stem cell fraction from our computing versus the population doubling time at each interval. And they all have that characteristic look. As the doubling time increases, the stem cell fraction goes down. Less stem cells, less doubling. And the reason I'm confident that we have algorithms is because we can linearize this data quite easily. And with the same conversion factor, we get these plots. So we have this algorithm now where if you put in the population doubling time, you get out this other factor, which is based on stem cell fraction. And we get these nice, fairly well-specific linear plots. They're not the same. They're going to be different for different stem cells and different for different conditions. But once we've got one, I can talk about this slide. We think this is the next generation of stem cell counting. So first we have to do a foundational AlphaSim test analysis like I've shown you. We've got to do one of those studies that takes 3 to 4 weeks. And then with that data, we will generate the population doubling time stem cell fraction algorithm. We'll determine what that is. like the ones I've shown you. And let me just say, the ones I showed you are actually done graphically. We're just working up to develop software to do that in a computed, automated fashion. There'll be a lot more statistical significance there. But once we have the algorithm, then we can just do it. Right now, we're at 72 hours. We can do a 72-hour growth study. And from that population doubling time, because of those conditions, we can get the stem cell number. It's going to be fast, so a few days as opposed to a few weeks. And it's going to be relatively inexpensive because there's not a lot of cell culture going on. There are some conditions, and we have to be wary of them. These algorithms are going to be very specific for conditions. They're only going to apply for the same type of stem cell, the same culture conditions, because culture conditions changes all those parameters, and we know that. And it has to be in the same range of the passage of the algorithm. And that range is actually quite wide, as I've shown you. It's over at least an order of magnitude of population doubling time. So I want to, before closing, just make some acknowledgments here. This is all possible because of AlphaSim Corporation, our partner in this, who does all the software development with us. BioCore has been really helpful. Murray Cross has really kind of supported us in allowing us to do these whole umbilical cord studies. Drick is another of our collaborators that we've done some of the umbilical cord hematopoietic stem cell studies. And the UMass Amherst core facility, we're in the voucher program, which allows us to use that cell culture facility for doing some of these hematopoietic stem cell studies as well. So the last thing I want to say, I really do think that at this point, with this technology, with greater use by this community, everything will change going forward. And so I want to thank you again for your attention and for your support, and I'd be happy to entertain any questions or comments now and for the rest of the meeting. Thank you.